Hello there and welcome to this Biology Mind A-Level tutorial on active transport. So just to recap what we did in our last lesson, we learned how to investigate transport across cell membranes using different models. We looked at how agar jelly composed of an alkaline solution and phenolphthalein can be placed in an acidic solution to investigate the rate of diffusion. So remember, our agar jelly contained that alkaline solution. Phenolphthalein was our pH indicator, so it made our agar jelly a lovely pink colour when the agar jelly was alkaline. But as more and more acidic solution diffused into our agar jelly, that neutralised our alkaline solution inside our jelly, made it more neutral or more acidic, and therefore changed that pink colour of our phenolphthalein to a colourless appearance. We also discussed how potato samples can be placed in sucrose solution to investigate water potential in plant tissue, and we plotted that on a calibration curve and had a look at how we can use our calibration curve to find out at what concentration our potato is isotonic to our sucrose solution. And we also looked at how to use Bisking tubing to investigate osmosis in solutions of different concentrations. So we have three key learning objectives for today. Number one, having a look at active transport. Number two, the role of carrier proteins. And number three, looking at another active process that we call cytosis. So endocytosis and exocytosis. So let's kick off by having a look at active transport. So we've met simple diffusion, we've met facilitated diffusion, we've met osmosis, we have not met active transport yet. And active transport contrasts big time with those other three transports across cell membranes that I just listed there. So simple diffusion, facilitated diffusion, and osmosis, because active transport requires energy. So it's an active process, as it says in the name. Whereas all our other types of transport across membranes, such as diffusion and osmosis, are passive processes. So this is osmosis here, and that's the passive net movement of water molecules down a water potential gradient, remember, from high water potential to low water potential. Now, our active transport in comparison is against a concentration gradient. And that's really important you remember that, because remember with our diffusion and osmosis, that was down a concentration gradient, so from high to low. But in active transport, it's against a concentration gradient, so from a region of low concentration, which you can see here, to a region of high concentration. And this is really useful because it allows cells to build up storages of substances that would otherwise easily diffuse out of the cell. So keywords to remember for active transport is against concentration gradient, requires energy in the form of ATP, and it's from a region, movement of molecules from a region of low to a region of high concentration. So that's against the concentration gradient. Now, active transport requires the hydrolysis of ATP to provide energy to move those molecules from the low to the high concentration. So this is because moving particles against a concentration gradient requires energy. So ATP provides us with that energy. Now remember, let's contrast that to diffusion and osmosis, which is from high to low concentration or high to low water potential, down our concentration gradient or down our potential gradient, which is a passive process. It does not require energy. But active transport is an active process. It requires energy. Okay, so that's our key difference between our active transport and our methods of passive transport. So now let's have a think about what sort of structures enable active transport to occur across cell membranes. So we have carrier proteins and protein pumps in the phospholipid bilayer. So every carrier protein is unique to each type of molecule that needs to be transported across a membrane. So a common example of a carrier protein, for example, would be the sodium-potassium pump. Okay, so very similar to facilitated diffusion, active transport often relies on these carrier proteins to allow for the movement of molecules across the membrane. Okay, so we've got a nice table here where we can contrast our different types of transport across our cell membrane. So let's kick off with talking about active transport, since we've been talking about that quite a lot this tutorial. So active transport, how does it transport molecules through the phospholipid bilayer? Well, it uses carrier proteins, so we can fill that in here. This is a really useful table to jot down in your revision notes so that you really understand the difference between these 4 types of transport across cell membranes. Is it an active or a passive process? We know it's active. It requires ATP to provide energy for the movement of those molecules against the concentration gradient. And as I just said there, the concentration gradient, we're moving against it. So it's from a low to a high concentration gradient. I'll just use L for low and H for high there. So that's the way that we're moving against the concentration gradient. Now let's contrast that to facilitated diffusion, which is also through carrier proteins, so there's a similarity there. However, the big difference is that it's a passive process. It does not require energy in the form of ATP, and it's down the concentration gradient, so from a high to a low concentration gradient. Now again, let's have a think about that with simple diffusion. So I'll do that in the same colour because we're talking about a very similar process as facilitated diffusion. The big difference is that simple diffusion involves smaller molecules which are diffusing through the phospholipid bilayer. They don't rely on carrier proteins to get them through. They're nice and small. They can diffuse through our bilayer. Is it an active or a passive process? It's a passive process. And which way is it moving along the concentration gradient? Well, it's moving down, so from high to low concentration. And finally, osmosis. Well, osmosis utilises aquaporins, which allow for the movement of those water molecules across the membrane. It's a passive process, so it does not require energy in the form of ATP, and it is also down at the potential gradient, remember, from a high to a low water potential. So this is a really useful table to jot down in your notes. Really, really try and remember that active transport is an active process. It requires ATP to provide energy for that transport of molecules against the concentration gradient from a low to a high concentration. Whereas these three types of transport across the cell membrane, they're all passive processes. And all that means is passive does not require energy. And because we know it's passive, so what does that mean in terms of which way it's moving along the concentration gradient? It means it's moving down the concentration gradient or down the water potential gradient in the case of osmosis. So from a high to a low concentration gradient, or if we're talking about osmosis, remember we always talk about the water potential. Okay, so that's our summary of our 4 different types of transport across cell membranes. So now let's look at a method of transport across cell membranes that we've not looked at yet, called cytosis. So cytosis is in the form of exocytosis, where exo means out of, or endocytosis, which I'll just jot down here, where endo means into. So why do we have cytosis as well as all of these other ways of transporting molecules across cell membranes? Well, we know that diffusion is mainly for small molecules. It doesn't rely on these other mechanisms of getting molecules across the cell membrane. Cytosis is for large molecules, so larger molecules have difficulty traversing a membrane. Now, interestingly, cytosis also involves energy in transporting those larger molecules. So here you can see we've got a vesicle, which is essentially, how I think of it, is it's a little bit of the plasma membrane that forms this nice bubble in the cytoplasm, and inside this bubble, or vesicle we should call it, is our large molecules that we want to move out of our cell. So our exocytic vesicle is going to fuse with our plasma membrane here, and it's going to allow the ejection of those large molecules out of the cell. And you have the opposite happening in endocytosis. Remember, endo for into, exo for out of. So in endocytosis, you get this sort of bubbling off of your plasma membrane to form a vesicle, and that vesicle now contains those larger molecules that were on the outside of the cell originally, now nice and trapped inside this vesicle, and that can then be transported into the cell. So let's look at that in a little bit more detail. So exocytosis, remember exo for out of. So this is when a vesicle containing molecules of substances, which you can see in here, fuses with the inside of our plasma membrane, and that causes those molecules to be secreted from the cell. So vesicle moves to the plasma membrane, fuses with that plasma membrane, and releases those larger molecules to the outside of the cell. Remember, it's an active process. And what does that mean? All that means is it requires energy. Okay, so that's exocytosis, exo for out of. Now, endocytosis, remember endo for into, so into the cell, moving larger molecules into the cell. It's essentially the opposite of exocytosis, so you have the cell membrane binding to the larger molecules on the outside of the cell in the extracellular fluid, forming a vesicle, which forms from the sort of in-pouching of that plasma membrane there, and this vesicle then allows large molecules to enter into the cell. So with endocytosis, there are two types of endocytosis that you need to know about: phagocytosis and pinocytosis. So let's start with phagocytosis. So phagocytosis is endocytosis, which allows for the uptake of solids, which you can see here. So, for example, phagocytosis is what macrophages do. Remember, macrophages are a type of immune cell. And when they consume pathogens, when they engulf pathogens and destroy them, that process is termed phagocytosis. And all that is the plasma membrane bubbling around this solid particle or a pathogen on the outside of the cell and bringing it into the cytoplasm of that cell. So it's a type of endocytosis. Pinocytosis is very similar to phagocytosis, except it allows for the uptake of fluids. So phago is for solids, pino is for fluids. Okay, and that's endocytosis. So we've covered active transport, we've covered two types of cytosis, exocytosis and endocytosis. So now let's do a little bit of a quiz to see whether that information all makes sense to you. So number one, how is the phospholipid bilayer arranged in the cell membrane? So give the video pause at this point and have a think about what you think the answer might be, and I will reveal the answer in a couple of seconds. So the answer is B, so hydrophilic heads face outwards. So remember, hydrophilic, water-loving, heads face outwards, and hydrophobic for water-hating, tails face inwards. So I'll just draw that out for you. This is our plasma membrane. So these are our hydrophilic heads, which are water-loving, they're facing outwards, and our hydrophobic tails are poking inwards. And we know that it's in that orientation because our hydrophobic tails are water-hating, they're hiding on the inside of our plasma membrane, inside that bilayer, whereas our hydrophilic heads are on the outside. Okay, next question. Which of these do carbohydrates not play a role in? Again, give the video pause here and I'm going to reveal the answer in a couple of seconds. So it is transport of ions across the cell membrane. So remember, those are channel proteins or carrier proteins, not carbohydrates that are involved in that. Whereas carbohydrates are involved in adhesion between cells in a tissue, they're involved with hydrogen bonding, and they're also involved with binding to hormones and antibodies at the cell surface membrane. For example, in the structure glycolipids, for example. Okay, and then which of these does not affect the rate of simple diffusion? So again, give the video pause, see if you can answer this. And I'm going to reveal the answer now. So the answer is the availability of ATP for transport. So why is that the answer? We know simple diffusion is a passive process, not an active process. Passive processes do not require ATP to provide energy. Only active processes require ATP to provide energy for the transport of those molecules against the concentration gradient from a low to a high concentration. So that means when we're thinking about what affects the rate of simple diffusion, the availability of ATP does not affect the rate because simple diffusion does not rely on ATP. If this was what affects the rate of active transport, then the availability of ATP for transport would be an answer. We know that the surface area of the membrane affects the rate of simple diffusion, and we found that out in our agar jelly experiment. We know the size of the molecule that is diffusing affects the rate of simple diffusion, and we know that stirring the solution containing the substance that is diffusing also affects the rate of simple diffusion, because if you stir the solution, you're going to maintain those concentration gradients and allow for diffusion to occur faster. Okay, so that's a lot we've covered there, well done. We've covered active transport, we've covered cytosis, and we've covered some exam questions. So well done for getting through till the end, and I will see you for the next tutorial.